Good morning, everyone. Today we will have our natural product session, and I first of all would like to welcome all of you, the audience, but also the speakers. And this natural product session was organized by the IUFAR Natural Product Committee, and we are very happy to have this session. It will give you an overview from basic targets of natural products to preclinical up to clinical studies. And you will see that natural products is a fast-moving field. And we will have, I will give my hand to Professor Spedding, who will introduce the first speaker.
Okay, thank you. Well, actually, it's a considerable delight for me to be able to introduce Professor Jean-Luc Wolfender, who has been one of my guiding lights since I was Secretary General of International Union of Pharmacology, because I believe that metabolomics is probably the best omics at present for our major issues. And Professor Jean-Luc Wolfender is very famous in the field of natural products. He's a chemist. He's also Dean of the Faculty of Sciences at the University of Geneva. His work is, for me, actually one of the key areas forward for natural products. And he's going to talk today about the combined target isolation and metabolomic approaches for the efficient discovery of potential therapeutic agents from natural sources. And this gives us a possibility of being able to deconvolute natural products and also to be able to effectively work out what's potentially active and what's potentially toxic. So, Jean-Luc.
Thank you very much, Michael, for the very kind introduction. So it's a pleasure to be here at IUFAR. And indeed, I will try this morning to show you a little bit what we do. Mainly, I'm a chemist, so I will show you mainly what we do on the technical aspects and on the way to apply metabolomics to try either to annotate compounds in complex mixture, herbal extracts, for example, but also use this technique in order to target the isolation of bioactive compounds for drug discovery. So, just a brief introduction on natural products. Natural compounds are looked at privileged structure. They have been shaped through evolution, mainly as defense compounds, but they are heavily used in traditional medicine, in herbals, for example, and this has proven their activity against many targets. And this is mainly related to their, chemical complexity, which allows very selective binding. And if you look at these natural products, you can summarize the diversity of this compound in a kind of chemical space. So all these dots will correspond to compounds. And from these slides, you see that if you compare natural products, for example, to compounds which were issued from combinatorial chemistry, you see that the space is much wider. And actually, this space matches quite well the one of drugs which are currently on the market. So that's why there was a strong interest to use natural products as a source of new drugs in general. And about 30% of all drugs that are currently on the market are still coming or are from natural product origin. And also these compounds also help to find new targets. Maybe they will not be translated into drugs directly, but they will inspire chemists to design novel active agents. And when you look at drug discovery, what you would like to have is pure and well-defined natural products. But the main challenge for phytochemists like us is that these compounds are indeed convoluted in a very complex matrix, which is a crude extract. And we develop technologies in order to profile this crude extract with a very high precision. And this helps us to either target the isolation of compounds of interest like this one, or also to standardize the extract to look, as this was mentioned by Michael, for bioactive compounds, active principle in phytopreparation, or looking also at potential toxic compounds. But for this, metabolomics is really needed. And having this deep knowledge on the metabolite composition give us a lot of avenue and possibilities today, because if you master the metabolite composition of all these herbal extracts, you can first start to make some rationale about their use in traditional medicine. Through isolation and pharmacological assay, you can prove the activity of some of these active compounds. And today, with the development of the digital era, you start to be able to build connection between all these metabolite profile, their possible bioactivities, and this field is really blooming, and I will try to show you how we start to make this connection on a semantic web way to combine experimental data with prior knowledge. So very classically in our field, when we perform this type of study, we start with natural extracts. We perform what is called the bioactivity guided fractionation, where we can follow activity of the extract constituents through fractionation with chromatography, obtain pure constituents, and do their characterization. So this path is pretty long, and because of this, most of the companies have refrained to do this natural product drug discovery, but they didn't really find very new chemical structure until now, and combinatorial aspects can help the development, but not really the discovery of new scaffolds. And in this process, what is important is to profile the extract to dereplicate known compounds and avoid their isolation. So that's how this technique starts to bloom in the field. And today, we are using especially LCMS for profiling the extract in a very comprehensive way. And this information are very useful because they give us a holistic view of the composition of extracts. And with bioinformatic methods, we can start to target the isolation of compound of interest. So it's really like finding a needle in a haystack with a powerful magnet, let's say. So to give you an idea of the complexity of the plant metabolome, we were interested, for example, to work on TCM drugs, which are extremely complex because that's a mix of many herbs. Here, 8 herbs, for example, for curing acute dermatitis problem here. And very strangely, if you profile the extract with quantitative methods like LC coupled with ELSD, you will see mainly one peak. And guess what is this peak? If I'm zooming, so when you start to zoom, you start to see other metabolites. But if you do, for example, an NMR experiment of the whole extract, that's the signal you will get. And actually, all this is related to sucrose. So that's, since this was a mix of roots of many medicinal herbs in China, of course, sugars are very important. And you see that quantitatively, there were mainly sugars. But there is hundreds of constituents where they are. They are sitting here. And if you profile now the extract with an LCMS method, you start to see them. And you can even go much more deeper. So that's a low-resolution chromatographic LCMS profile, but you can do it with very high-resolution aspects. And I know that Michael liked that because this is a way to deconvolute the extract. But this is just to show you how complex it can be. All these peaks correspond to different compounds, okay? And with LCMS, you can really do this at a very, very highly sensitive method. So that's what you will ingest when you take a TCM. So what are the toxic, what are the beneficial compounds? So that was just to show you on a technical viewpoint what we could get. And these profiles look like that if you have this coupling with LCMS. So basically, all these peaks correspond to compounds in the extract. And the methods allows you today to do this. I think I will switch it here because it's more easy like that. These methods allows you to detect not only the main compounds, but also very minor constituents at the same time with very untargeted methods in metabolomics. And basically, the information that you get, of course, is not directly the structure. What you will obtain is the molecular ion, from which you can retrieve the molecular formula. And automatically, you can retrieve what is called an MSMS spectra. And this is a fingerprint for the metabolites, and you can query this fingerprints against databases. The main issue with natural product chemistry is that these databases are not very extensive, and there is more than 300,000 natural products which have been published, but we have to find strategies in order to do this search within silico methods. So basically, that's the type of information that we retrieve, so molecular formula and MSMS spectra. I will summarize this with these icons in the slides to come, and they can be looked at a first glance as a fingerprint of the molecule. Just to give you also a glance of the sensitivity, you have seen the resolution of LC. The mass spectrometry today is extremely sensitive and has a very high dynamic range. So it's a little bit like if you were having a high-resolution camera flying over the Amazona, and your high-resolution camera allows you to see the leaf on the trees, for example, but also all the mushrooms below the trees, and even the insect grazing on the mushroom below the trees in the forest. Okay, so that's just to give you an idea on how sensitive can be the mass spec data. So sometimes you look at some metabolites and they are at this level. So you have always to have this in mind when you do this type of profiling. So in our group, we try now we work with several thousand of natural extracts, and we try to do this in-depth metabolite profiling in order to retrieve as much as possible chemical information to target the isolation of pure natural product, let's say in a targeted manner, and then assay only the very interesting molecules in bioassays. So for this, it's very difficult to interpret all the spectra. So we take the advantage of what is called molecular network. So molecular network is a method which allows you to make connection between metabolites in a given LC-MS profile. And this is done by looking at the similarity of the spectra of each of these molecules. And at the end, you will see that we are able to annotate a very large majority of all these metabolites. Have in mind that annotation by mass spec is just an approximation of the structure. It's not the full identification, but it gives to the chemist already a lot of information, and it helps also standardization of drugs and quality control. So basically for each of these dots, you will see that we can retrieve an LC-MS spectra for the molecular formula and its corresponding MSMS spectra. So if you look at a real extract, this is how it looks like here. This is a real profile of a given plant extract. Well, this was Arabidopsis thaliana in this case. And on this dimension, you have the mass spec, and here you have the chromatography. So every dot basically will correspond to a given metabolite. Sometimes you have more dots than metabolites, but to have a rough idea, that's how it looks like. So that's really the complexity of what we are looking at. And this is for a single plant. And for each of these dots, you will get the MSMS spectra. So you can get more than 1,000 annotation for a given plant extract. So if you look at TCM, it's even more complex, but that's just to show you how deep we can go into these data. So in order to annotate the metabolite rather correctly, it's very important to have a knowledge of what was published before, and for this we developed this Lotus initiative during COVID time, and my PhD and postdoc here, they were able to retrieve all publicly available chemical structure, and they make a link with their biological taxons. This is very important because every structure which has been published has to be linked to the source organism. And this taxonomical information helps you later on to do the correct annotation because, of course, we have to take into account the biosynthetic background. And this database is freely available on the web. It's here. And I think it's a very important resource for probably all those which are working on natural products because this helps you to make rational on the chemical composition of a given plant, for example. And you will see that we can link this also with pharmacological activity. So for example, in this Lotus database, we could query for any plant. So you can do a query like that. So the data are now in Wikidata, and we are just, because we could put them in Wikidata, which is a way also to keep them almost forever. But so our database now is querying Wikidata with the taxon data that we put in. And for example, you can ask, what are all the metabolites present in Arabidopsis thaliana? And this will display this type of graph where you see in which database you have the metabolites. And you can do this with, by writing this type of query, but I'm not very good at doing this. But today, you can ask ChatGPT, and you can say, write me a query to ask for retrieving all chemical structure from Arabidopsis thaliana, and it works. He wrote me this query, I run it, and I could retrieve 600 natural products reported for Arabidopsis thaliana. So, and this is also coming in the field. We will have this natural language interfaces to interrogate our experimental data. So, in Lotus, we could retrieve 700,000 pair and about 250,000 natural products. And this is the chemical space of Lotus. So every dot here will correspond to a metabolite. And through this way of displaying the compounds, they will be displayed by chemical similarity, you see. And this is a way to classify these natural products. And all of them will have the link to their biological organism. So all this information now is available to you on the web. And this is quite useful because for us, for the annotation aspects, we can now use all this chemical space because we have all the structure. And from this, we can calculate, and it's possible to do computation for this. This is starting not to work. OK. We can calculate in silico MSMS spectra. So this is predicted fragmentation spectra of each of the molecules that you see in the chemical space. And now, with this, we can query our experimental data and do annotation. Of course, this type of query is not perfect because this is not experimental against experimental, but it's quite useful. So we have different ways. I will not go into details to compare this MSMS spectra against either experimental data here or against in silico spectra. So our strategy is mainly to build up this huge library of in silico spectra because also the condition may change from one instrument to the other, and systematically do this annotation with this strategy. So now we get the MSMS spectra for each of the molecule, and I'm going to show you how through molecular network we can make these links. So have in mind that every of these peaks will get an MSMS spectra. If the MSMS spectra is similar, that means that the structure are very likely to be similar. And by using this, we can build up molecular network. So a molecular network is a way to display any of the ions which have been detected, so the molecular weight ions, if you want, and in a kind of spectral space. And if a given molecular ion is given an MSMS spectra, and if another one in the same extract is given another MSMS spectra, if these two MSMS spectra are similar, and an algorithm can calculate this, then they will be displayed in the space very closely. And this will generate cluster. So you see, for example, here we have a cluster of molecules which are chemically similar because they exhibit similar spectra. Okay, so this is one structural type. And if you do this on an extract, you will have several structural types. So you will end up with a display like that, where you have different clusters. And every cluster will correspond to a given chemical structural type. So this is quite nice, because in an extract, you will already know what are all the major class of compounds. And you will know that you have analogs which can be connected by this way. So just to show you that it works, so we could generate, for example, here, the metabolite profile of about 300 U4BAC species. They are quite interesting, especially for anti-cancer activity, for example. And we could compute, can you start the video for me here, if you click on it? It's running, it's not running maybe, yes? I'm looking at the guy, but it's not running anyway. No worries, but what you would have to see here is that by calculation, you can start to build up clusters by MSMS comparison. and at the end, you will get this cluster for the whole data set. So after calculation, you will see all these different clusters, and this will help the chemist now to know where is a given structural type and annotate all the compounds. So this is for the 300 extracts, so we collected about 1 million spectra for this data set. And now I can show you what are the type of information that we have, for example, in one of these clusters. So for each of the dot will represent one molecule, if you want. This is not completely true. There is many dots for one molecule, but have this in mind. Then for every dot, you have this MSMS spectra. We can do then the spectral similarity score for each of them. And now we have to annotate this compound. So in this case, for example, we had one standard, one pure compound, which was a 4-bol ester derivative. So this one was fully identified. We know that since they are connected in the cluster, that means that all the other dots should have similar structure. Okay, but what are these structures? So for this, we run our algorithm, which is comparing the MSMS spectra against in silico data, and the system will give us candidate structure for each of the dots. So we will have several candidate structure with a given score, and this is what we call the annotation. Okay, so this will annotate mostly all compounds, but of course with a given score. So we are never sure of the exact structure, but this is already quite a lot of information. In order to improve this score, we will use the information from Lotus. I'll show you the database, because here we have all the taxonomic data. And this will help us to sort out what are the correct candidates for a given structure, because we can take into account the biosynthetic pathway. So by taking into account the taxonomic distance of the organism, then we can re-rank the structure, the top 5, for example, and have a better confidence in the annotation. That's our strategy at the moment. So this is really improving the annotation score. Just to show you this, for example, on one of these clusters, for this ficus species, for example, in the first round, we were having this annotation for different flavonoids, but we were unsure, for example, of this one and this one, because such type of aglycone, for example, were not reported in ficus species, but when we applied this algorithm, we could correct this structure. They were still having the same mass, very similar MSMS pattern, but based on this, we could do the correct annotation. So this is really helping as a filtering method. So finally, if we apply all the tools that I show you, we were able to annotate quite a bit of this metabolite in this cluster. And they were matching with compounds reported in U4BAC. So we know, for example, that this cluster were really containing interesting 4-bol esters. I will show you later on what was the activity of this compound. And this was also proved because we could get known standard, which shows us that we were on the right track. The other information which is very interesting is that for every node, every mass here, we have the distribution of the mass across the different samples. And we can see, for example, that this one is shared between different extracts. And then it's very interesting because with this, you can make correlation, for example, with bioassay results. So just to show you also how it looks like on phytopharmaceuticals, you probably all know ginkgo biloba. I'm trying to use it a little bit to improve my cognition. I don't know if it works really, but that's anyway what I'm ingesting, okay? And if you look at the metabolite composition of ginkgo biloba and you apply our methods, if you look, for example, at all the peaks which are detected at 5% threshold, you will get about 84 annotation. And that's mainly corresponding to what was published on ginkgo biloba. But if you go down in the forest to see the insects grazing on the mushrooms, you see that we end up with about more than 1,000 annotation for the same ginkgo biloba extract. And this is real compound. This is not noise. I mean, the metabolites are there. So I know that Michael is always asking me, did you detect some pesticides? So maybe they can be in this very low background. And that's anyway things which are taken by people. So the main question is what threshold is meaningful. That's really an issue for us. But that's just to show you that we can go at these 2 different levels. And I had a PhD who ended up last year who actually get an award just yesterday in Dublin. We were having a conference over there. And Adriano developed a method which allows us now in metabolomics to distinct what is the major metabolome and what is the minor metabolome. Because in mass spec, it's extremely sensitive. You will detect a huge amount of things. So we are trying now to use detectors which are generic detectors which give a kind of semi-quantitative response, which is not the case with mass spec. And we use these methods to filter the DMS data in order to look at the major metabolite if we want, or only at the minor one. But that's an information which is very important. And he developed a very complex algorithm which allows you to deconvolute every HPLC peak to understand what were the ions behind. I will not go into details. But basically, when you profile a plant with this, on the top trace here, you have this semi-quantitative trace, which already show quite a lot of constituents. And on the bottom trace, you have the MS part. And by applying this filtering, we could now go down from thousands of features to about a few tenths of main constituents and a few hundreds of minor ones. So this is something which starts to be workable and well filtered for annotation purposes. So we are really at the beginning of this part. But I think this type of information between major and minor metabolites are very important, also from a toxicological and pharmacological viewpoint. So now, I show you only chemistry. I'm a chemist, so that's why. But I still have to show you that we can perform some bioassay on all these extracts. So at the moment, we are trying to also screen the extract with different assays in collaboration. And we try to make connection between this molecular network and bioactivity. And you will see that this allows us to prioritize interesting compounds. So, for example, if I took back the example of this U4BAC set that I showed you before, all these extracts were also assayed. They were assayed, so that you remember, that was our molecular network. And now we have all this metabolite profiling data. And these plants were assayed for Wnt signaling pathway in collaboration with Vladimir Katanaev. And you are more specialized than myself on Wnt signaling, but it's quite interesting for carcinogenesis, and we can find different compounds which are modulating this pathway. And in our case, about 20 extracts showed a very good inhibitory activities on Wnt. And we were trying now to make a connection between these 20 extracts and the 300 that we were profiling. So for this, we took again our molecular network, which is here on the right, and we color code all the cluster according to the bioactivity we get at the extract level. So all the ones which were in blue were inactive, and in red they were active. So you see, mainly on the whole network, it's mainly blue. But then we could really look at the level of every cluster if we were having red color or orange color. So we had metadata to every of these dots. And this allows us to finally select 21 clusters among 2,300 for the whole data set, which were clearly linked with bioactivity. And I will just show you one of these clusters, how we could deconvolute this cluster. So you see there was several metabolites which were linked to bioactivity here. And I can just compare on the same plant clusters which were inactive. So the inactive cluster were showing very different type of structure, but the active one were indeed from the 4-ball ester type, which was quite correct also based on prior knowledge. But of course, with mass spec data, we didn't know exactly the structure. In order to find the real correct structure, even if we have annotation, you need to isolate and go for NMR. And that's the work we do also in the lab. And what we develop on our side is that we have this very precise metabolite profiling data, and we are able now to translate this at the analytical HPLC scale to find out where are the compounds of interest based on mass spec. And now we can go to a semi-preparative scale to follow the same compound and isolate them for full de novo identification. So we don't do any bioassay guided fractionation. We can just do a cherry picking of the one which are of interest. And we can do this at small scale. Urs knows this guy, Emerson. So this is the pope of isolation in my lab. So Emerson works at different scale, you see. HPLC scale, flash chromatography, and even MPLC. Just to show you that if you need to isolate hundreds of milligrams of compounds, you need to go on this part. In India, probably I would need this to get standards, for example. So basically, with this approach, now we are able, after isolation, to get the correct structure of these compounds, and through, of course, interpretation of the NMR data. And very interestingly, in this case, we pop up with this 4-ball ester. And if I look at the structure of this guy here, actually, the structure of this 4-ball ester was very similar to tigilanol tiglat, EBC46. And this, we found it in a euphorbacea plant from New Caledonia. And this one was found in an euphorbacea from northern Queensland. But the one on the right is known to be an activator of PKC, and our was a potent Wnt inhibitor. And the one on the right actually underwent phase 2 clinical trials for you see, a given type of cancer here. So these diterpenes are very promising as an anti-cancer agent. So that was a proof of concept that we could really cherry-pick some compounds, and we end up with very strongly bioactive compounds. So I show you this for 300 extract, but we can work at a larger scale. This was not complex enough. I have to go a little bit further. So we have the chance to have the collection of 15,000 plant extract from the Pierre Fabre collection. And at the moment, they are not doing any more drug discovery on this, but we can profile part of this, about 10% of the whole collection, with the method I show you. And this gives us access to the chemical space, or the virtual chemical space of all these extracts. So now we try to make some queries, but you see, it starts to be extremely difficult. This is the molecular network of about 1,600 extracts, and it goes further. So it becomes impossible to visualize the data. And our idea is to convert this into a virtual chemical space for querying and using artificial intelligence algorithms to help us to do decision here. So how to do this? Well, this is really a complex metabolomic set, and you know that people working in metabolomics like to find needles. So I'm not finding needles myself, but I have a very nice crew of people who are developing tools to look at the needles in this very complex haystack. So the limitation that we were having is that with molecular network that I showed you before, we cannot visualize the data anymore. Because for any of the dots in any of the cluster, we have many annotations. So how to find a way to work with this? And we are reproducing this metabolite profile 1,600 times, and we have still 15,000 extracts to look at. So it's quite a big work. So how to deal with this? Actually, and this is very new, we try now to convert our experimental data into a semantic web format. And actually, this is how the web is organized. If you look at Wikidata, this is semantic web data. And the idea, and this is, a few groups are starting to do this, is to convert your own experimental data in the same format, because then you can do expert queries. So the idea here was to deconvolute any of these extracts. You remember the dots correspond to molecular ions. Every dot will have an MS, an MS-MS. Every dot will have different structure in spike code. They will belong to a given LCMS spectra. And on the top of that, and this is most of interest for you, there is a biological activity. Okay? So now, you can build up what is called a graph, a knowledge graph, and it uses this RDF format. So this is how the semantic web is organized. Just to show you an example, you probably know this lady. Okay? And so actually, Meghan Markle is the subject, and Prince Harry is the object. Don't tell Prince Harry that he's the object of Meghan Markle, but they are linked through a predicate. That's how it works on Google, okay, on the semantic web. And there is also information. For example, Meghan Markle is played by Rachel Zane in Suits. I never follow Suits, but it seems that is the case. So now if you do a query in Google and you say, who is the husband of the actress playing Rachel Zane in Suits, the answer will be Meghan Markle, but you see that there will be some relation with Harry, but also with his previous husband, Trevor Ingleston. So that's just to show you how you can query the web, and you can query the data the same way. That was mainly my message here. Okay, so forget the very complex computational aspect. We develop many tools now which are putting together structural annotation, also prediction of spectra, linked to different databases, and we can query our data in this way. Just to show you how they look like, this is now all the different features, the LCMS data, their annotation. Every annotation has a spectra, so that's how the graph information are. And I will not go into details, but if we deconvolute completely all this information, every object is linked to the next one as a triplicate. And if you do this on these 1,600 extracts, and especially if you link this, and I think this is coming now, with bioactivity, because every of the plates containing the extract has a given bioactivity, the information is there. You need to query and see how you can make your connection. So we have done this, and now we have about 40 million triples in the database. And so everything is connected, okay? What is interesting for us is that now you can connect bioactivity and extract composition. So a given extract has a given composition, and you can start to make query to see if statistically there is some of the compounds which can explain the bioactivity of the extract. And it's quite powerful because the data are organizing in this semantic web format, and the web is organized the same way. So you can start to make links between the platform knowledge with the web knowledge. So for example, we are starting, this is really new on our side, we are starting, this one is not working anymore, to make a link with ChemBL, for example, where a lot of pharmacological information are embedded. But through the structure, we can start to make these links, and we can generate hypotheses, okay? So we have been doing this, we start to do this on this data set, and now the idea is we cannot look at all the metabolites, but all the metabolites explain one extract. And now every dot that you see here is the whole spectral information of one extract. So every dot is one extract, 1,600 extracts. And we use algorithm to see if extracts can cluster together based on chemical composition. So for example, here with this visualization algorithm, you see that we have two clusters here as well. And these clusters, for example, here highlighted in green, were the active extract of the same data set for antiparasitic activity. So we were having 7 extracts out of the 1,600 which were active in this case. And they cluster together. So now we have to know why they are clustering. Is there any features which are common to this extract which can explain the bioactivity? Okay? And actually this was the set of plants which were highlighted this way. And taxonomically, they are not related. So there should be some chemical elements which are common. And we can do queries. So we can now start to write Sparkle queries, like on the web, on our data. And for example, here, we look at the rate of annotation. So we ask to our graph DB, what were the chemicals which were more frequently annotated in the active extract compared to the whole data set. And we end up by looking at the class of rotenoids here. So there was much more rotenoids in this extract which were annotated than the whole data set. So now, with this, we can make the assumption that indeed these 7 extracts have rotenoids inside. And indeed, we could, by looking back at the data on the mass spec side, we could indeed find traces of rotenoids in all of them, mainly these 2. And actually, in this case, we could even order these 2 to prove that they were inside. So, and these are known also to be rather toxic molecules. But interestingly, at least in the assay that we perform in Basel, they had a very interesting selectivity index, which was a bit puzzling, but at least this could have been toxic molecules. But you see, this is just a proof of principle. We could get this just through the statistic information, and straight away we highlight this compound. At the moment, we could isolate all these ones, and we are checking if there is really a selectivity aspect. So that's the beauty of plants. You can get a whole series of compounds, and you can check if there is any structural activity relationship, at least in this assay. So for us, this was a proof of concept, and now I will finish with this. You remember, this was the chemical space of all knowledge data from Lotus. Okay, that's what I started with. And now, with the metabolite profiling method I show you, we start to be able to annotate. And on the 1,000 seed extract from Pierre Fabre, which were really biodiverse, in blue, you see all the annotation that we could get. So you see that already with this 1,600 plant extract, we cover quite a large part of the chemical space, which is known to date. There is parts, for example, here, which are not covered at all. Why? Well, this is mainly coming from the extract, because Pierre Fabre was doing extracts which were targeting molecules in a given log P range in order to be druggable. But for example, here you can have, for example, glycosides, which will not be considered as druggable from this rule. So that's explaining why we have some holes. But this is showing also that even with a small set of plants, we can start to have a rather high chemical coverage. So now we try to use all our annotation as a virtual chemical space, and we are really at the beginning of using this data for doing artificial intelligence-guided isolation. The very final things I would like to show you is how we can link this data with ChemBL. So for example, this is what you see here in blue is all the annotation that we retrieve from Pierre Fabre. And in yellow, what you see is all the structure that we have in ChemBL. So so we were looking at IRA in this plot where we were having both dots which were in ChemBL and relationship with metabolites in our data set. So here on this branch, you see these two compounds, and that's the structure of these compounds. And they were interesting for us because they were reported to have a rather interesting IC50 value in an assay against Leishmania donovani, which were also screened in Basel in collaboration. And we were interested to know what were the natural products related to this one, which were in our data set. So we found the ions. So if we know the ions, we can find in which plant they were belonging to, and based on the mass and the retention time. So finally, we identified two plants where these compounds were reported, or analogs, and we could target the isolation of these compounds. So we could retrieve finally, about 8 or 9 of these terpene analogs to those which were bioactive in Campbell. And we isolate this with the targeted isolation that I showed you before. And we could retrieve some interesting activity for some of the derivatives, just to show you that this gives us also a possibility to explore analogs. So either we explore analogs or we explore parts which are completely uncovered. That's a little bit the idea. So with this, I hope that I could convince you that we are at a point where with metabolomics, we start to be able to do very detailed metabolite profiling of the extract, bridge this with taxonomical information, create virtual chemical space for queries, and use also semantic web data to make a relationship. And I think it's important not only for drug discovery, but probably if we evolve with this method, we should maybe start to have possibilities to make relationship with phytotherapeutical herbals and maybe understand a little bit better, or at least have clues on how this is working. So, of course, very importantly, this has to be linked to bioassays. And hopefully, at the end, this will really be useful to help the targeting of pure natural product or the chemical composition assessments of herbals. So with this, I would like to acknowledge all the people in my group. This is a rather old picture, but you see they were very dynamic. So, and we showcase ourselves recently on a chemia paper in the form of a molecular network. So that's all those which were mainly involved in this, the development of the tools I show you. We are in Geneva, near from the Geneva fountain. Caroline is with us, so sometimes she's in the same building as we are. And we have the chance to have this NMR and mass spec facility, so we are very open for collaboration with all of you. With this, I would like to end up, but before just ending, I have to advertise you also a little bit, because yesterday I was in Dublin at a GA meeting. So the GA is the Society for Medicinal Plant Research and for Medicine Plant and Natural Product Research. I think the activity of GA are very resembling to what we are doing here at IUFAR. IUFAR is probably much more pharmacologically and toxicology oriented. We are more chemically oriented at GA, but we were discussing also with some of you last night. It would be interesting to maybe bridge some of the activities of our societies. GA is very open for this, and I would just like to advertise you that we will have next year in Krakow our joint meeting, which is a very big meeting for all natural product chemists, and it would be interesting to have also more pharmacological talk and toxicological talk related to natural products. So please be welcome to come in Krakow, and thank you very much for your attention.
Absolutely marvellous. When I became Secretary General in 2014 of IUFAR, one of the key things that I wanted to push was evidence-based science for natural products and their identification. So Jean-Luc was therefore one of the key factors of that, and you can see how his work's developed, which I find extraordinary. So, questions, please. Dr. Bikash?
Yes.
When we look at metabolite, and we look at how many are active metabolite, so when we take the product into regulatory pathway, so many regulators, they said that you have to identify at least 4 active ingredient and some metabolite. And can you make it a point, like you said that you look at the chemistry part. So when you go for further, so if the active metabolite is the pharmacologically active, and at the same time, if you have a mechanism, like you said about artificial intelligence, can you predict how many of them can develop an ADR also at the same time?
Can develop, sorry?
Adverse drug reaction or adverse event.
Yeah, so this is, of course, extremely challenging, but indeed, that could be one prospect. So indeed, for the regulators, I think, well, depending on the country, they are asking for at least being able to characterize a few metabolites, usually a few. It's like 4 in India. In China, I think they are asking 1 active principle per plant if they do a formulation. And you see that if we go really in depth, we can annotate hundreds. So we don't have to show this too much to the regulator as well, because they might ask for more. but the point is that, so on one side first, we have to, I think I show you very quickly this minor and major metabolite classification for the industry, for standardization. I think you can use our tool with this filter first to look at the major metabolite, because anyway, not maybe for toxicology, but at least for pharmacological effect, they are the more likely to have an effect. So we get the structure indeed. So from this, if you identify the active principle, this has anyway to go through isolation and some bioassays. And you know that in herbals, we don't have always the active principle. So if we don't have, you can use it as a standard, as a marker, taxonomic marker for the plants. But indeed, your question was if you have the active principle through maybe virtual metabolization, can we predict the toxic aspect? I think starting from the structure, because what we are just trying to deliver is structure. If there is any program which allows you, for example, first to predict metabolization, and they exist, so you can predict all possible metabolites for a given structure, phase 1, phase 2. And from this, you can probably, if any data would exist somewhere on the web on these metabolites, or maybe analogs, you can start to make links. But we didn't do it, but I think, yeah, potentially it's possible, provided that the structure is correct.
Just quick query. It makes a lot of difference, like
Yeah. You can, yeah.
It makes a lot of difference when you go to the clinical part.
Yes.
Like if you look at a giant in India, like Himalaya, Natural Remedies, they usually collect in a particular time, like in the rainy season, summer season, and that activity is, it is huge differences there when you go for a clinical efficacy. So is it database when you're going to create the data like you had done, is it going to reflect the diurnal variation or in terms of, collection for the rainy season or other season, and where you can see that a lot of differences there in terms of efficacy in a clinical trial?
Yeah, no, that's a very good point. So what I show you were mainly static data, so metabolite profiling at a given moment. But indeed, a plant is not a plant. That means that all the biochemistry reacts to the environment, and it's well known that moment of collection will change the metabolite profile. So you can use the same methods to profile this at different time of the year, for example, and this is done in metabolomics, and observe the changes. And indeed, in future, if hopefully everybody can share metabolome data, this information should be provided as metadata to know if this metabolite profiling was done in which region at which timing, and this will start to give clues. For example, on the ficus data, we were with the Malaysia. I just show you one cluster with the flavonoids. This was the purpose of the study. So we analyzed about 80 of these plants at different timing, different location.
Okay. Two very quick questions, and then go on, but please quick.
Okay, Tanya Garkovic, NCI, and I have a practical question. I'm also a chemist. My question is about time. So you've shown lots of beautiful UPLC traces going from 30 to up to 120 minutes. I'm assuming you're also hitting multiple collision energies in your MSMS spectra. So how long did it take, not only to generate the 1,600 plant extracts, but also to process them? Because I'm assuming your data files are huge.
Mm-hmm. Yes. So for the profiling of 1,600, we went to shorter gradients than the one I show you. So usually we profile about 100 plants over a weekend. We do 8-9 minutes gradients. So the acquisition is about 100 plants in 2 days for the acquisition. Then the processing is indeed a major issue. At the moment, for the 1,600, to compute all the annotation, it took us about 3 weeks of CPU time. But now we are mainly privileging this filtering that I show you with semi-quantitative data. And the idea is to have a completely fully annotated report of about a few tenths of main metabolite, and this can be done in a few seconds. So, but this is just there. We are published. The last one is not yet published. We submitted. So this will give a rationale for this part. But we can discuss about that.
Okay. Go on, quickly.
Okay, so thank you for your presentation and good work. As you talk about the molecule network, I think it's very important for us to understand the compound and how to analyze the bioactivities for the extract based on the network, molecule-based network. This is the one question. Another is, as you talk about the mine dates. But when we mine the dates, we want to find some active compound or active extract or fractions. The question is which one? The pure compound, a single one, or with the complex of compound is our goal.
Okay, so
Thank you.
so yes, so with the network to find out the active. At the moment, we are doing this connection with many extracts. So we use the high number of extracts to make our correlation, and you have seen that we can find out that a given ion can be found in many extracts. So we use statistics based on a high number, and based on this, we can say, okay, most probably, since this ion is always popping up in different active extracts, it should be the one. So that's how we could say it might be the one. But of course, in sometimes you need to have fraction data, and at the end you need to have the pure compound. So that was for the first question. For the second one, indeed, when working with herbals, we all know that it's very tricky to find only one active principle explaining the whole clinical effect of the herb. You have seen with TCM, for example, there is many compounds. So at the moment, we can only offer a way to identify and assess the activity of single compound. But of course, synergistic effect has to be taken into account. Metabolization have to be taken into account. So there is still a big gap between chemical composition and clinical efficacy. But by building this knowledge, maybe, and looking also at results in general, maybe finally we could get this type of links to be done.
Okay, I'm sorry. We have to
Something
stop
that we
there.
can discuss later on.
Yeah,